Thanks so much for joining me for another edition of Parker Biology. Today we'll be recapping cellular respiration. It's important for us to remember that no matter how an organism obtains them, we all require organic molecules. So if we look at the plants in this picture, we know that they are autotrophs or producers, and they're making their own glucose or organic molecules. Whereas the chimpanzee in the picture is a consumer or heterotroph, and he needs to take in these organic molecules. This diagram reminds us of the interrelated nature between photosynthesis and cellular respiration. Through photosynthesis, organic molecules and oxygen are produced, and the mitochondria then are able to use those organic molecules and oxygen, releasing CO2 and water, but also producing ATP to help power cellular work. We also need to keep in mind oil rig, that oxidation is loss and reduction is gain, and we're still focused on redox reactions because throughout cellular respiration, we're going to see electrons being transferred. When it comes to this electron transfer, it's really important that we see electrons moving in small steps, and we see a gradual transfer. What this does is maximizes a controlled release of energy so that ATP can be synthesized. If hydrogen atoms and oxygen atoms came together all in one big step, we'd see a really explosive release of heat and light energy. This would be really uncontrolled, and we wouldn't be able to harness that energy as well as we can in cellular respiration using more of a stepwise transfer. In looking at the aerobic processes associated with cellular respiration, we know that the mitochondria is a key organelle. In terms of its structure, it does possess double membranes, both an outer membrane and an inner membrane. The inner membrane here is shown in blue, and we can see that it is highly folded. And all of these folds, or cristae, serve to increase the surface area, which tells us some pretty important processes must be taking place across that membrane. The space between the outer and inner membrane is referred to as the intermembrane space, and the far middle of the mitochondrion is called the mitochondrial matrix. This is a great diagram to come back to remind ourselves of the key processes associated with cellular respiration. We'll be starting out in the cytosol, looking at glucose being broken into two pyruvates through the process of glycolysis. We'll see that if oxygen is present, the pyruvate will get to enter the mitochondrial matrix, where pyruvate will be oxidized into acetyl-CoA. Acetyl-CoA will then enter the Krebs cycle or citric acid cycle. The main event here is electron carriers will be generated, NAD and FAD, who will carry electrons over to the electron transport chain. We can also see from this diagram that there are a couple of ATPs produced both at glycolysis and through the citric acid cycle, but the main source of ATPs is going to be through oxidative phosphorylation as a result of the ETC, or electron transport chain. This diagram also hints that there are two main ways to make ATP. In glycolysis and the Krebs cycle, we can see that ATP is generated by substrate level phosphorylation. And what that really tells us is that the phosphate group is being transferred from another organic substance. It's being taken from someone, or substrate level is stealing a phosphate, as we can see in this picture. The phosphate group was attached to another substrate, but it's being stolen from there, added to ADP, or adenosine diphosphate, resulting in ATP, or adenosine triphosphate. If substrate level phosphorylation is stealing the phosphate, oxidative phosphorylation does use the power of oxygen and the reactions of the electron transport chain. This is where we'll see an inorganic phosphate, or P subscript I, added to ADP. If it's not ripped off of someone else, if it's just a free-floating phosphate, that'll be oxidative phosphorylation. The first step of cellular respiration is going to be glycolysis, which literally means the splitting of sugar. This is the process by which glucose will be broken down into 2 molecules of pyruvate, and this does happen out in the cytosol. There are 2 main parts to this. We're going to see energy investment and energy payoff. The cell has to spend 2 ATPs in order to release 4 ATPs, resulting in a net gain of 2 ATPs. Also through this, in addition to some substrate level phosphorylation, creation of those 2 ATPs, we're also going to see some NADH generated. We'll see some electrons removed from glucose and passed on to this vital electron carrier, who's going to carry those electrons off to the electron transport chain. Glycolysis does occur whether or not there is oxygen present. Here we see the steps in more detail. We're not responsible for these details, but it is nice to see that there's a series of 10 steps, each catalyzed by its own individual enzyme. Notice that we do have these 2 ATPs being input, 4 ATPs being released, again for that net payoff of an overall 2 ATPs gained. We can also use this diagram to see why we refer to this ATP production as substrate level phosphorylation. If we notice, there's a phosphate group on this intermediate. However, after its conversion, there is no longer a phosphate group there. That phosphate group is actually removed from this organic substrate and added on to ADP. Same thing over here. Phosphoenol pyruvate did have a phosphate group. That phosphate group got stolen and glued on to ADP. So both of these are substrate level phosphorylation because that phosphate group is stolen from someone else. Just to come back to that initial roadmap, this reminds us that out in the cytosol, glucose is broken down into 2 molecules of pyruvate through the process of glycolysis, generating a net gain of 2 ATPs via substrate level phosphorylation, also resulting in removal of a couple electrons by an electron carrier, NADH. Following glycolysis, if and only if oxygen is present, pyruvate will enter the mitochondrion. Now, prior to the citric acid cycle, or the Krebs cycle beginning, pyruvate has to be converted into acetyl-CoA. And that's going to be taken care of by this oxidation of pyruvate step. We'll see removal of a CO2, addition of coenzyme A, resulting in acetyl-CoA. This is the little baby step that has to link between glycolysis and the Krebs cycle. Once pyruvate has been converted into acetyl-CoA, acetyl-CoA can enter the citric acid cycle or the Krebs cycle. And as it moves through this cycle, for each molecule of pyruvate, we'll see 1 ATP, 3 NADHs, and 1 FADH2. If we started with a glucose molecule, though, remember glucose was actually split into 2 pyruvates. So for every 1 glucose, we'll see 2 turns of the citric acid cycle. Now with that, the ATPs are generated through substrate level phosphorylation, and the NAD and FAD are important because they are electron carriers. That acetyl-CoA is going through further breakdown, and as it gets broken and broken and broken apart some more and more electrons get removed, and those electrons will be carried off to the electron transport chain by these electron carriers, NAD and FAD. Again, we're not responsible for the nitty-gritty details, but we can see acetyl-CoA coming into the cycle, a series of enzymes converting a bunch of reactants into products. We've got this biochemical pathway, a chain reaction going on. And we can see that some CO2s are going to be released. And here we can see these electron carriers, NAD and FAD, along with some substrate level phosphorylation. That ADP is stealing phosphates from GTP, stealing phosphates substrate level. So yes, it's nice to see that CO2 comes not only from the pyruvate being oxidized into acetyl-CoA, but CO2 is also being generated through this citric acid or Krebs cycle. And that's nice to tie back to the equation, and we can see why we breathe out CO2. But the main importance of this cycle are these electron carriers. The electron carriers, NAD and FAD, are carrying electrons off to the ETC, or electron transport chain, located along the inner membrane, or the cristae, of the mitochondrion. So these electrons are going to be bounced down a series of mostly protein components, and we'll see a whole series of oxidation and reduction. We've got redox reactions as we see them accept new electrons and pass them off like a game of hot potato. Now, part of why this is so efficient is the final electron acceptor, that highly electronegative oxygen molecule, waits at the end of the chain, luring the electrons to bounce towards it, ultimately resulting in formation of H2O, or water. This picture is one way to imagine this, that NAD and FAD are dropping off electrons to the electron transport chain, again embedded in the cristae. And as the electrons bounce, 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 they're being pulled by that final electron acceptor, oxygen. When protons unite with these electrons, that forms hydrogens. And as the hydrogens and oxygens unite, the resulting molecule is water. Another way to visualize the electron transport chain is here, looking at those cristae, that inner mitochondrial membrane. And again, we see NAD and FAD dropping electrons off. We can follow the electrons, bouncety bounce, 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 down the ETC. And with each bounce of the electrons, we see them releasing some energy. The energy released from the bouncing of electrons is used to pump protons. And these protons are going to build up in the inner membrane space. We get a really steep proton gradient built up. And once we see a steep gradient, we know that the likely movement of these protons is going to be from the area where they're highly concentrated to an area where they're less concentrated in the matrix. Now, the only way the protons can come back through, since they're highly charged, since they can't just wander through this cristae, this membrane, the protons need to diffuse back through using this embedded protein channel, this ATP synthase. And as they do, ATP synthase is going to spin, picking up ADP, inorganic phosphate, generating ATP. Notice that this is an inorganic phosphate, so this is no longer substrate level phosphorylation. This is a great example of oxidative phosphorylation, relying on the presence of that oxygen to pull the electrons across the chain. Again, when the H's and the O's get together, there's that water. So bouncing down the electron transport chain, building up a proton gradient, and allowing those protons to diffuse back through. Using that gradient to perform some cellular work or produce ATP is referred to as chemiosmosis or even proton motive force. Hopefully then, this reminds us of the breakdown from glucose to 2-pyruvates. If oxygen is present, pyruvate entering, being oxidized into acetyl-CoA, acetyl-CoA taking a couple of turns through citric acid cycle, resulting in a whole bunch of electron carriers, bouncing electrons, pumping protons, spinning ATP synthase, and ultimately a whole bunch of ATP to power our cells' work. Thanks for joining me. for another edition of Parker Biology.